Okay, so now let's move on to the TCA cycle. TCA stands for tricarboxylic acid cycle, which reflects the first product in the TCA cycle, which is citrate. It's also called the Krebs cycle after Krebs, who discovered it, and it's also called the CAC, which is the citrate acid cycle. So we're going to start out, we've discussed pyruvate. Pyruvate being converted into acetyl-CoA by the pyruvate dehydrogenase enzyme. And now we are going to take acetyl-CoA and put it into the TCA cycle. And this is going to be done through a condensation reaction with oxaloacetate. This reaction will generate citrate. And the enzyme that does this is citrate synthase. Now, important to note here that this reaction is irreversible in the body. It's irreversible because recall that this bond between the coenzyme A and the acetyl group is a high energy bond. And so this is a very exothermic reaction, having a very high negative delta G, making this a one-way irreversible reaction. The other thing to note is that acetyl-CoA is a 2-carbon molecule, oxaloacetate is a 4-carbon molecule, and citrate is a 6-carbon molecule. So we are adding 2 carbons into the cycle, forming 6. So that means we're going to be losing 2 carbons in order to regenerate the 4-carbon oxaloacetate. Now the next step of this reaction is to generate isocitrate. The enzyme that does this is a aconitase. This enzyme has a delta G around 0, which means it's readily reversible depending on substrate and product relationship. The next reaction we're going to talk about is the conversion of isocitrate into alpha-ketoglutarate. The enzyme that performs this reaction is isocitrate dehydrogenase. Now, being a dehydrogenase enzyme, this is going to be a redox reaction. So in this step here, we are going to convert NAD to NADH, transferring electrons from our isocitrate to the NAD carrier, and we are going to release our first CO2. So here we are taking ourselves down to a 5-carbon molecule. The isocitrate dehydrogenase is not a reversible reaction, so it has a very substantial negative delta G, making this not reversible. Now the next step is we're going to take our alpha-ketoglutarate and we are going to generate succinyl-CoA. Like isocitrate dehydrogenase, this is a redox reaction and it's going to be mediated by alpha-ketoglutarate dehydrogenase. So in this reaction, much like the previous one, NAD will be our acceptor of electrons to form NADH. And here we are going to lose our second CO2, bringing us back to our 4-carbon intermediate. Being a dehydrogenase reaction, this also has a very large negative delta G, making it also irreversible. Now at this point, the CO2 that is being evolved from these two reactions can diffuse into the blood, be picked up by red blood cells, and as the red blood cells are releasing oxygen, they can pick up the CO2 so that carbonic anhydrase can convert the CO2 plus water into proton plus bicarb ion. This then travels, of course, to the lungs, where the reverse reaction occurs, releasing CO2 for expiration. Now the other step in this reaction here of the alpha-ketoglutarate is we are going to add a coenzyme A. This is going to essentially raise the energy level of succinyl-CoA to a higher energy state because this is a high energy bond. What that will allow us to do is in the next reaction harvest this energy as we generate succinate from succinyl-CoA. We are going to harvest this energy in a slightly different step, and that is coupling phosphate to GDP in a high energy GTP bond. The energy of this CoA addition, release of this bond, of that high energy, will drive this synthesis. This enzyme is called succinate thiokinase. Now at this point, the succinate is going to be prepared for the final couple of reactions. So we are going to convert the succinate into fumarate. Now fumarate is very interesting because it has a double bond. We're preparing it for the final steps in the TCA cycle. To introduce a double bond, we are going to need a special redox system, and this is going to convert FAD to FADH2. This is done by the enzyme succinate dehydrogenase. And this enzyme is unique that it is embedded in the inner mitochondrial membrane. Of course, it is facing the matrix of the mitochondria, so it has access to the TCA cycle intermediates. But the FAD is bound to the enzyme. So these electrons that are transferred to FADH2, the FADH2 does not leave the enzyme. It stays coupled to the enzyme. That being the case, and the enzyme being in the inner mitochondrial membrane, these can be released in order to regenerate FAD by releasing these two electrons into the electron transport chain, specifically at the level of coenzyme Q. Now the next step in this reaction is to create the next intermediate, which is malate. The enzyme that does this is fumarase, and this is simply a hydration reaction. So we're just simply going to add water across those double bonds that we created in fumarate to generate malate. Malate finally will be processed into oxaloacetate. This is done by the malate dehydrogenase enzyme. Being a dehydrogenase, this of course is our final step of energy extraction, and we will be converting NAD to our carrier of electrons, NADH. And in doing so, we will be oxidizing malate into oxaloacetate. Now, curious, this forward reaction has a delta G that is equal to positive 7.1 kilocalories. So the question would be then, if the forward direction has a positive delta G, meaning an endothermic reaction, where does the energy come from to drive this reaction forward? The reverse reaction, of course, has a delta G of negative 7.1, so you would think it'd be more favorable this way. But in skeletal muscle, this reaction here, this condensation reaction between acetyl-CoA and oxaloacetate, is happening very rapidly because of the large amount of acetyl-CoA coming down the pipe from glycolysis, because skeletal muscle's need is to just generate energy. So that being the case, the concentration of oxaloacetate at any one time in the TCA cycle is very low. So if we remember our thermodynamic equation, notice the product over the substrate. This is our key factor in this. This is the free energy change of the reaction, and this is fixed. This is a constant depending on this reaction. Our gas and temperature, of course, are constant. Now the natural log of the product over the substrate. So if the product is very low, and in this case oxaloacetate condensing with acetyl-CoA will keep this concentration low of oxaloacetate. Substrate is high because we're having lots of processivity through the TCA cycle, so the concentration of malate is very high. So this would be oxalo over malate concentrations. This would be a very, very small number. Small number here, the natural log of a number much less than 1, is a negative number. So because of this product to substrate relationship, this will be a large negative number that will generate a negative delta G to drive this reaction forward. This high delta G will become very important in the liver because we are going to take advantage of this reversibility in this reaction, and malate only in the liver, not in the skeletal muscle. This will be used for producing glucose in gluconeogenesis. Regulation, very simple in these reactions. We have a dehydrogenase enzyme here. We have another one here. And we have a final one here. These dehydrogenase enzymes are regulated by the NADH to NAD ratio. What this simply means is that these dehydrogenases need NAD in order to perform the reaction, and the higher concentration of NADH is inhibitory. The other way to look at it is NAD is depleted when NADH ratio is high, and so this is inhibitory to the dehydrogenase enzymes in the TCA cycle.